All right, so thank you all very much for coming back. I hope you had a good lunch. To follow on our hippocampus morning, we have, I was trying to think of something that is similar between the two, and basically it's how the brain deals with our interactions with other people. So I don't know how you say that in one word. Afternoon.
Social neuroscience.
Social neuroscience afternoon. There you go. Hippocampus morning and social neuroscience afternoon to make our brain big. Okay, so our next speaker is Dr. Michael Tarr. Dr. Tarr is co-director of the Center for the Neural Basis of Cognition and Cowan Professor of Cognitive Neuroscience in the Department of Psychology at Carnegie Mellon University. He has received the prestigious Guggenheim Fellowship as well as the NIH Eureka Award, which sponsors exceptional high-risk research. Ever since his PhD at MIT, his work has been the center of many debates about how human visual systems function. His work began with a novel explanation of 3D object recognition and later moved to understanding human face perception. His now famous little characters called Greebles, which he has used to help us rethink whether or not human face perception is specialized, have become a mainstay of cognitive psychology experiments in vision. His more recent work has focused on trying to understand the intermediate layers of visual processing using some real-time fMRI methods. Today, we'll be given a tour of the architecture of human face recognition by one of the world's foremost experts. Please join me in welcoming Dr. Michael Tarr.
So a couple things. One is I'm going to give a bit of review here because I'm not sure everyone necessarily knows some of the old data. So I'm going to talk about data, actually, some of which is collected by a colleague of mine, Marlene Berman. Some data which was really from the lab of one of my old students, Isabelle Gauthier, who's a professor at Vanderbilt now. Some other students of mine, Adrian Nestor and Junxia Kyung, and then John Piles is a postdoc with me right now. I'd also say that I like to have conversations while I talk, which means that if you have a question, please do interrupt. That's fine to do. And if we don't get through everything because we got into some interesting digression, that's great as far as I'm concerned. So, all right. There's a basic fact about understanding face recognition in humans, and to some extent what's been done in neurophysiology in monkeys as well, which has sort of been in the field for a long time now, and it's probably the most prominent single piece of data that people have about how face recognition works, which is that if you have people or monkeys look at objects and you have them look at faces, and then you look at where in the brain there's selectively higher responses for faces above objects, there's this area of the middle fusiform gyrus, particularly in the right cortex, in the right hemisphere of the cortex, that's more selectively active for faces as compared to objects. And this was originally done in PET by Justine Sergent in Montreal, and then Nancy Kamwisher later on did the same thing in fMRI. And this has been dubbed the fusiform face area, or the FFA, and that's what I'm going to start with here. I'm going to talk about that. I will just mention that there was recently an article in the Scientific American on their website, if I can read the whole article here, which was published on April 1st, which would be a clue to what this article is about. It was entitled, "Neuroscientists, we really don't know what we were talking about either." Okay? And this is a paragraph from the article, which I'm going to read to you, short paragraph. "People who read a lot of neuroscience news have probably noticed several consistent contradictions," says Laura Solkis of Dartmouth College. Some students say that different brain regions work in concert. Some studies say that different brain regions work in concert to perform a single complex task, whereas other studies argue that a particular cognitive function, such as recognizing faces, is basically the sole domain of one region. The thing is, just because a part of the brain shows more activity than another, it doesn't mean there's only one piece involved. But it's just so easy to pick a neglected area, dress it up with some colorful fMRI studies, and present it to the world as a distinct functional region of the brain. How can we resist? For neuroscientists, the brain is like the world during the age of exploration. We have major continents named, but there's plenty of room for new countries. Everyone wants to plant a flag. Okay. Now remember, that was an April 1st line, but I almost could have written that in all seriousness about this kind of result. So often the best humor is something where there's actually some truth to it. And I just want to emphasize that this isn't a theory. And fMRI is a rule. Anytime you show a particular kind of activation associated with particular tasks that people do, that's not a theory. It's a piece of empirical data that might contribute to a theory, but you really would want to understand what's actually happening in all sorts of different kinds of ways about why that might be true. That's even modulo all the issues that may exist in terms of the fact you're actually measuring blood flow and not neural activity directly. But given all of that, I want to talk about this region and other regions that may be related in terms of face processing. And this is the other issue, and this is actually a comment maybe about all of brain science, but particularly about, I think, the study of high-level vision. So we probably know something about early vision, okay, V1 through V4, I would say. And then we talk about things like object recognition and place cells or place representations or face representations and so on and so on. And how you get from something that looks like an oriented receptive field in V4 to neurons or regions of the brain that code for something like faces is really then a miracle occurs. I don't think we really know, and there's no honest, good theory of how vision works as you move up into higher levels. So we've got a lot of work to do, and hopefully, hundreds of years from now, we might be able to say something meaningful. But so here's an outline of what I'm going to talk about. So first question I want to ask is, what's driving FFA? Let's ask these are some older studies we've done about what's really driving FFA, and Chris alluded to it already, and I'm going to go through some of the other issues as we go through. But so what's driving FFA? Well, one way to ask that question is, what's special about faces? Why should there ever be a situation where faces seem to recruit some piece of neural real estate more than that same piece of neural real estate is recruited for other objects? All right, so one of the things that's particularly unique about faces is we're very proficient at discriminating between individual faces. And for most of the other stuff in the world we have to deal with, we don't have to worry about being that proficient at telling apart individuals within that class. So as long as you know it's a chair, you can sit on it, it doesn't really matter what the particular model of chair is, and so on. And the idea really is that for faces in particular, we become very proficient and automatic at being able to tell apart things within the class. And the other thing about faces that really makes them special is that they have a high degree of similarity within that class. So compared to, let's say, all the dogs in the world or even all the chairs in the world, faces have basically the same kinds of parts or features in the same basic configuration, and yet we're very good at pulling them apart and telling based on those parts and those configurations exactly what the individual exemplar is. Okay, so those two properties seem to be special for faces, and by special I mean they're unique task demands that we seem to satisfy in doing that, in doing face recognition. But what if we acquire proficiency for telling apart other stuff where there's similar kinds of constraints, highly visually similar, and we become quite proficient at it? I'm going to quickly talk about 3 different domains. You alluded to Greebles. Here's a Greeble for any of you who have not seen a Greeble before. How many of you have seen a Greeble before? Wow. Okay. That's a little scary, actually. But these are Greebles. I'll talk about that. Birds and cars, BMWs in particular. So what we do with Greebles, and this is old work again with Isabel. By the way, Greebles are the only stimulus class I know of that has their own Wikipedia page, which must be something. But they're these synthetic objects, and we train people to recognize these things so that they were very proficient at them. And that means that they could name each of them as individuals, they could do same different discriminations and so on, and they could do it quite automatically and easily. To show you how good the training was, there was one of our original subjects from the original study, which was run in '97, I saw in 2006. And I gave a version of a talk where I had Greebles with some names on them. And he came up afterwards and he said, "Those names are wrong." And I said, "What?" And he said, "Oh, no, that's not Jepu, that's Pimu." And we just reversed the label somehow in the slide I'd done. And he still remembered the individual Greebles, almost 8 years later. Okay, and it's about 7,000 trials of training. So the question is, what happens in the brain when you learn about Greebles? Okay, well, so here's the brain of Greeble novices. And this is when a Greeble novice is looking at faces. And this particular contrast here is a contrast between an upright face and an inverted face. And that recruits a standard piece of neural real estate in middle fusiform gyrus associated with face processing. Okay, you compare upright Greebles to inverted Greebles, you don't get anything happening in that particular region. Okay, you make people Greeble experts, you still get responses in their same region in middle fusiform gyrus for faces. But now for your Greeble experts, you're also recruiting that same piece of neural real estate for Greebles, right? But only post-training. And this has been replicated in various different labs over the years. There's some debates about failures to replicate and things like that. If you actually look closely at why they failed to replicate, it usually has to do with changing task demands or changing the stimuli or doing a variety of other things. But it's certainly been replicated multiple times. Okay, so what about other kinds of stuff? Well, here's a lovely study that Isabelle Gauthier ran. What she did was she took bird experts and people that were really hardcore birders that go into the wild and look at birds and name them all really carefully and collect them in their little books. And then she also took, in this case, it was BMW experts. So they really knew about how to tell apart different models of BMWs in all the different years quite proficiently. And turns out you never find a birder who's also a BMW expert or vice versa. I don't know why. you know, but they're not going out in the M3 apparently and then looking at birds. But the car guys, and it was all guys, to be clear also, recruit, when they're looking at cars, the same region of middle fusiform gyrus, and the birders looking at birds recruit the same region of middle fusiform gyrus. Again, this is right hemisphere. We've got radiological right here. And then you look at the birders when looking at cars, and there's not much going on. They don't seem to really care. And you look at the car guys when they're looking at birds, and there's not much going on. They don't seem to care. Again, above and beyond other kinds of objects. So the same kind of data that we saw for the Greebles holds true for birds and cars. OK, now here's another study that's actually run in my lab several years later. If you look here, what we've actually got, we've got a really extreme set of birders. We had people in this set that were birders that had been doing it for 40 years, where they were going out every year and looking for birds long periods of time. And I'll show you why this data is particularly interesting. We get the same effect that Isabel got before, which is our non-birders show very little selectivity for birds, but our birders show high selectivity for birds in the mid-fusiform area. But if you look at the face responses, and this is the strange part, you've got your non-birders that are showing this typical response that you see for looking at faces, but then look at your birder group when they're looking at faces. Okay, so it's a bit of a puzzle. I'm going to get back to them in a second, but I thought it's worth showing right now so you can start thinking about what's happening. And by the way, to be clear, these birders are perfectly proficient at recognizing faces behaviorally. It's not like they're prosopagnosic in some form or impaired as far as we can tell. I mean, some of them might be a little bit odd, but that's a different issue. Okay. I had one talk where I gave this talk and someone said, they're all autistic. And that's why they can't recognize, why they're not showing this response, but they're not. They really aren't. So what are some implications of all of this first set of data? Well, I think category selectivity for faces, the kind of thing you see when you talk about the FFA, is really a concatenation of task demands, not a product of faces qua faces. And what do I mean by task demands? Discriminating between exemplars within a class, exemplars that are distinguished via configural information in particular. So it's really the spatial relations between common kinds of features. So those two things I think that are special to faces can be special for other kinds of things like cars or birds or Greebles. Okay. Other object domains can with acquisition of expertise. And I don't like the term expertise so much because people tend to then call the FFA the expertise area as an alternative theory. But that's at least as vacuous a theory as calling it the face area. Okay? You have to say, what is it about expertise? What are you acquiring? What are the particular components of that? Impose similar kinds of task demands, which is a critical part of it. But you can say that the neural architecture that's supporting face recognition, at least from this kind of data, is domain general. It's not specific to one particular class of objects. And I think the other thing we can say is that as you become proficient in doing any of these tasks with any of these domains, you become much more automatic at doing it. So you do these individuation tasks without thinking about it, without consciously trying to do it. OK. So returning to our outline, what drives FFA? I've sort of just summarized that, this proficiency at fine-grained discrimination. And the next question we want to ask is, what's changing with experience? And this gets back to that slide I just showed you about the birders and their absence of responses for faces. OK, so here's some more detailed data from our original Greebles study. And one of the things which we hadn't really thought about at the time, but in retrospect we should have thought more about, is that it turns out as they're acquiring proficiency for getting better at Greebles, this is the neural response for Greebles in the red bars, those blue bars are the neural responses we're seeing for faces. Okay? So actually, even in our original study, as we train people up to be Greebles, they're actually getting lower neural responses for faces. So it's very similar to what we're seeing in our birders. Quite interesting. Okay. This is training a particular single prosopagnosic. He was an acquired prosopagnosic that had a closed head injury. SM, one of Marlene Berman's patients. And he actually, we were training to become proficient at Greeble recognition. He, again, was highly impaired, if not entirely impaired at face recognition. And what we saw, actually, was that over the course of training, so this is pre- and post-training is what these bars are. And these are different, you know, my eyesight's gone from turning 50. I can't read anything far away anymore. I can read it if I step back. This is faces as compared to Greebles. This is faces compared to objects. And this is faces as compared to fixation. And in all cases, the response for faces is going down Even though he's highly impaired at face recognition, any residual response he has for faces is going down with training of the Greebles, and the Greeble responses are going up. So, and then you can actually see that in the neural data pre and post for him as well. We actually stopped the training as soon as we realized this, because we were worried that whatever residual ability he had for faces might be being damaged somehow by doing this training, which is a little bit of a scary thought, even though he seemed 100% impaired at face recognition, whatever he had left. So again, it's the same pattern we're seeing. Here's the strangest set of data. These are a bunch of birders, some of the data I showed you from Junqia's thesis. And this is basically showing you across 4 different tasks, and you don't have to really worry about the tasks. There are different perceptual judgments. We're measuring how good people are at birding. So that's what this behavioral expertise score is. It's how good they are at telling birds apart. Okay? And then here, we're measuring the strength of the neural response for birds as compared to objects. And by the way, this is very regional. We use Rhode Island birds. And if you used Asian birds, they don't show this pattern. So it's not about birds generally. It's really about Rhode Island birds. And you can see that as the subjects that are better or more proficient at telling birds apart behaviorally are also the ones that show the highest neural response. So this just replicates the kind of thing that we've seen multiple times before. Okay, here's the data for those exact same subjects in terms of their neural responses to faces. Okay, so the better they are at birding, behaviorally measured, the lower the neural response for faces. It's pretty striking. And this is, again, multiple tasks, so we're really sure that it's not just some idiosyncratic aspect of one particular kind of processing stimuli. Question?
I'm a little bit confused about what you're measuring here.
Oh.
I mean, with respect to
This is
response to faces, when you show a reduced correlation, couldn't that simply reflect like a generalization processing in that area?
I'm not sure what you mean by generalization processing, but we're measuring the summed activity in this region of interest, the right midfusiform, and the strength of that activity for that particular contrast, which is in faces minus objects, for instance. And so I think you're presaging exactly what I'm going to say about what's happening. So if I didn't, get back to me and we'll talk about it. But right, so but for that region that's been associated with face processing, the striking thing is that the better you are at birding, the lower that neural response actually would be. Which is kind of interesting given other theories of what supposedly this region is doing. And that forces you to think more carefully about what it really might be doing, which is the basic point here. So this is just showing in a completely different study with different birders, we replicated the effect again. So it's a pretty robust effect. That, again, the better you are at birding, the lower your neural responses to faces, but not your behavioral recognition of faces, your ability to recognize faces. So what is going on here? Okay, so this is somewhat speculative because you're just basing this on the nature of these correlations and how they were and how they were measured. But the argument is that fMRI is always measuring the summed activation over millions of neural units, whether it be at the level of single neurons, columns, whatever it is. We don't exactly know what the neural code is like there, but that each of these units, at some level, what I'm presuming is that it instantiates some kind of feature. And in particular, what I think is going on in FFA is it's instantiating the features of individual exemplars within the class. And there's some evidence coming out of Girl-Spector's lab and some other labs, and actually Gautier's lab, now that's exactly what's happening. When they've done fine-grain, high-resolution imaging of FFA, you'll see that it seems to be coding individual features within the classes that you're proficient at. Because it's coding all these individual features, and you only have one, let's say one domain that you're usually proficient at if you're a face expert, but you haven't done anything else, that's going to be overrepresented across that whole network. As soon as you learn a second domain, some of the units get shifted from being devoted to face features to being devoted, let's say, to Greeble features. So now, just because of the way fMRI works, the summed activation over that whole pool, the ROI, the region of interest that you're looking at, is going to be decreased. So it's not that the birders, for instance, are getting, is this answering exactly what you were, yeah, good. It's not that they're, in some sense, losing any ability to recognize faces or any other resources. It's just that faces are the only things in there for people that aren't proficient at birding, presumably. As you get more proficient at some particular task, some of those neural resources get used up on something else, and when you sum it all together. So it does show you one of the weaknesses of fMRI, that you're always summing over these large numbers of units, making assumptions there's uniformity. When there's not uniformity, you get effects like this. The other point I'll make, which is interesting about how these features might be organized is that Grosspector's work and more recently the Gautier work is suggesting that you do have some fine-grained selectivity for lots of different kinds of objects that you're not actually proficient at telling apart. That it's there, it's not really strong, it's not nearly as strong as it would be for a domain like birds or cars where you're an expert, or for faces certainly, but it is there. And for instance, in the Grosspector paper, they found some evidence that there was selectivity for sculpture, and I think for cars in a case where people weren't car experts. And you say, well, why would that be? But you can imagine to the extent that you're instantiating features that are going to be critical for telling apart individuals within a class, you should also be pre-learning at least some of the statistical regularity of the features for all the classes that you know about. So that if you ever begin to work on individuating them more, and you're beginning to get more proficiency at doing that particular kind of task for that domain, those sort of latent potential is there, and you can begin to draw on that. So that there's some representational space that's much larger than faces or cars or birds or whatever happens to be most obvious when you do the neuroimaging. It's really covering the space of probably all objects, but that those features tend to be fairly generic for a class until you begin to hammer in and learn that class well. So I don't know if that's clear to people, but so it isn't just about these features. There's probably going to be some here for everything that you know about. They're just going to be much more generic. Okay, is that people with me? Good. So that's something about the impact of experience. So again, my argument is that FFA encodes features of specific exemplars, and I'm going to give some more data that sort of supports that argument. And then there's a question of what the functional role of this is. Why is it doing that? Like, what's its role in the face recognition system? And remember, I gave that quote at the beginning from the April 1st Scientific American article where they said, well, obviously, it's not a single piece of neural real estate. It's doing a whole cognitive function. And I think that must be true. It must be, around here, I probably don't have to tell anyone that it must be a network of mechanisms that are always participating to do any given task. But the question is, what's the functional role of this particular region in the larger network that might be subserving face recognition? So this is some beautiful work that Marlene Berman and her students Galia Avedon and Sibu Thomas and other students in her lab did. To be clear, I wasn't part of this work, but it falls in with the story so nicely, and it's fascinating work. So it turns out, probably about 10 years ago, people began to realize that there was a population of people that were prosopagnosic, meaning that they couldn't individuate faces, and they were prosopagnosic from birth onwards and never acquired the ability to tell apart faces. Okay, they're called congenital prosopagnosics, or I think there's another term that's sometimes used. We sometimes use the term face blind, is more in the popular press. And I think 60 Minutes did a piece 2 weeks ago with Nancy Kamwisher and Brad Duchesne interviewed about this. But it turns out it's a much more common syndrome than people realized. Now Oliver Sacks has suddenly decided that he's congenitally prosopagnosic too, which I'm not so sure about. But Jane Goodall definitely is. And there's a lot of other people that are fairly well known that are. And then if you look in a normal population, you can find people fairly easily. I'd say in a room of 100 people, you usually find one person. I don't know if there's anyone here that is, but it's not that uncommon to be either entirely impaired or very poor at face recognition. It's not naming. It's being able to visually tell apart the faces. That's the real syndrome. And it's not also that they can't see faces. They see them fine. They can see the emotions. They can tell the gender. They can tell you lots of other things about the face. They just can't tell two individuals apart based on the same kind of cues that we use. Now, they can use specialized things. Like if you're in a room with a couple people, you know who the people are, you might say, well, Bob has the glasses, Mary has bright red hair, and you could do it from that. So it's not that you can't use some local cues, but you don't effortlessly just tell apart the faces in the way we naturally would. So there's a population of people. Oh, and the last thing that's interesting about this group is it runs in families. So it turns out that you'll find, you know, two uncles and a nephew and a sibling that are all congenitally prosopagnosic in a family together. And there's some genetic work being done to try and understand, obviously, you know, what might be carrying this. But it's very common. It runs in families if you see it. So, so here's what Marlene did. She was imaging the congenital prosopagnosics. Here's your controls. And what you can see is right fusiform gyrus being recruited for faces more than objects. Typical result that we see time and time again. Very robust. So this is a control group. But look here at our congenital prosopagnosic group. I believe this is a population of 10. They've actually got some probably lower activation in other parts of the ventral pathway, but their actual activation for the midfusiform gyrus for faces compared to objects is completely normal. There is no statistical difference at all between the fMRI data you get for running a congenital prosopagnosic at faces compared to objects and running controls. So one of the things that tells you right away is whatever this region is doing, it certainly isn't sufficient for face recognition, because these people can't recognize a face to save their life. Okay. Actually, one of the most famous quotes about this is there's a person who talked about if they went to pick up their daughter at daycare and they had changed her clothes during the day because kids get messy during the day, he couldn't find her because he didn't know what clothes to look for anymore, because he was coding, oh, she wore a red shirt and now she's wearing a blue shirt. And so he'd have to ask them which kid was his daughter. Okay, so that's how profoundly impaired they are. And yet, when you have them look at faces, it's completely normal. So that's kind of intriguing. So what's going on? So Marlene, working with several people, had an idea that there was really a network of processes, which again isn't unique to them, but they were trying to articulate what that network of processes would be that's involved in face recognition. And looking at the whole literature and trying to sum up some of the things that are out there, people talked about core regions for face processing. And there's an idea that there's an early visual region I'll talk about a little bit more. It's sort of a little bit of a precursor and more occipital regions, precursor to the lateral occipital complex, flowing through into the fusiform area, the mid-fusiform area. We'll talk about STS as well as maybe being part of that network or not. But then you have information flowing from posterior to anterior regions, particularly into anterior temporal cortex, which has often been implicated in sort of identity processing, lots of semantic information about the person, okay? And then also into much more interior regions, anterior frontal cortex, where it seems to be much more involved in any kind of ambiguity and trying to sort out who the individual is, perceptual ambiguity. So there's the idea that these are the core regions involved in face processing. So what Marlene did was she used an early version of diffusion tensor imaging, which I'm going to talk a lot more about in a little bit. But basically, diffusion tensor imaging is a way of looking at structural connectivity in the human brain in terms of the white matter fiber tracks that connect one region to another, the long-range fiber tracks. Okay, so as opposed to the kind of structural image you usually see, which is looking just at the sort of gray matter structure, you're actually looking now at these connective fibers, which you don't usually see in this kind of typical MRI scan. And they were interested in how the connectivity of these different regions played out when you looked at congenital prosopagnosics as opposed to controls. And that's what diffusion tensor imaging would give you. So here's their data. Okay, so if you look at the DTI, and you look here are their controls. And they have this particular tract, I always get the name wrong, it's the inferior frontal occipital fasciculus, IFOF. Okay, this particular tract, which really connects sort of more occipital and mid-fusiform region into anterior regions, probably this region over here. That tract is normal in all of the control subjects, and they were all age-matched controls. Okay, gender and age match. Okay, and then here are the congenital prosopagnosics. And you can see in every case, there's degradation in that tract. And there's a little, this person's got some intact, but for the most part, some of them don't have anything in that tract at all. So, and they actually had a control track, the IOF, and the IOF track, which is the other track which goes, I think along in here, is completely intact in the congenital prosopagnosic. So it's not like they have some kind of white matter degradation all over the brain. They actually have fairly normal looking white matter. It just so happens this particular tract is entirely or almost or partially missing for the population of subjects. Moreover, they found that behaviorally, to the extent that they had any spared face recognition abilities, it related and it was correlated with the amount of spared fibers within that tract. So this person, I think in particular, was the best at face recognition out of the population that they ran. I think that person's actually a professor. Is that? No, there's someone who's a professor at Carnegie Mellon who I actually knew and didn't realize that he had this impairment. I think it's this person. So it's quite striking. So what this is telling you, you think about it, is that, first of all, normal FFA is clearly insufficient to support face individuation, which is striking because there's lots of things out there, not so much within psychology, but lots of other things out there in the popular press and other literatures have got interested in this. I had a huge argument with Ned Block one time about this. He kept insisting FFA was the place face recognition was done. I'm saying, no, look at this data. But it isn't sufficient. And really, in particular, connectivity, this network for anterior brain regions to these more posterior regions is critical for proficient face recognition. Okay. What is it Hillary Clinton used to say? It takes a village. So you have this whole network of processes. And if FFA is normal in general prosopagnosia, it's very intriguing too, because it means that if I believe that FFA is instantiating face features, these individual features of, for individual faces, whatever's happening there, how you're learning them and how congenital prosopagnosics are learning them must be relatively unsupervised, because the connectivity to the regions that you would think of that would provide supervision aren't there. Now, it's possible that supervision is coming from some other connectivity into that region that we haven't mapped out, but it is striking along the part of the ventral pathway that was assumed to be involved in face recognition. You've basically got that part of connectivity severed, and yet they're still learning somehow the right kinds of features for face recognition, even though they can't do the task. So I almost think of FFA as being sort of the unsupervised learning component of a system that ultimately has some kind of supervised component farther up within the stream. But it's learning the statistics and the individual features of the task, and they do it by looking at faces, even though they never can tell the faces apart. All right, so and that's all presuming one thing which we don't know, which is the FFA activation in congenital prosopagnosics really normal or not? And you'd have to do a lot more studies with them manipulating various kinds of stimuli to see how normal that response is. Like, it might be that it degrades in different ways and is reduced in different ways relative to controls. So there's still an open question about whether it's really normal. It looks normal under the most basic conditions, but there's more to do. Okay. And then the argument in particular also is a configural mechanism to put all these features together that ultimately allow you to say in a configural way that this is Mike and not Bob, because you've got all the features in the right places. That assembly must be happening downstream at the point of the more interior regions of the brain. So those are the basic arguments based on that. I think it's fascinating data. There's a really lot more to do about trying to explore whether or not the activity that we're seeing in FFA is really normal or not, and that may be very informative about the kinds of computations happening in FFA. Questions at this point? Okay. So in terms of talking about a larger network, we can say that FFA is certainly functioning in tandem with interior nodes. It's not living on its own, and the computations with this network, we're not completely sure, but I would argue that my theory of vision is basically you have filtering, unsupervised learning, and then supervised learning. I think that's a theory of everything. But that certainly here what we're actually seeing is that sort of an unsupervised statistical learning of the kinds of features in the domain that you're becoming proficient at seems to be intact, and I think that's probably what's happening there. Okay, so what about the functional roles within this network? Can we say any more about what we really think is happening in terms of the computations? So one of the things I've tried to do in my career is, because I don't think we have great models within psychology for the study of high-level vision. In terms of when I say great models, I mean models that make specific quantitative predictions. I've tried to take models from computer vision. Because the nice thing about computer vision models is, and this is what you were suggesting last night, right? That they take the same input, pictures of the world, and they ultimately often have the same goals, which is to recognize scenes or objects or faces or whatever it is. So presumably some of the principles by which they end up getting from A to B, and to the extent they get to A to B at all, not all these models work that well, but within machine vision, might be useful for informing some of the things we try and understand about how biological vision works. It at least provides a carefully thought out quantitative model that might be useful for testing against what we're seeing when we look at the neural data. So this is a particular computational model developed by Shimon Ullman. And he actually is one of the sort of true believers of the sort of David Marr idea that you really can meld computational approaches and things like machine vision with biological modeling. So he's tried to do that with this model. And this model is a model in which they use mutual information as a way of computing the image fragments that are best for doing a particular task. So what that basically means is, let's say you were interested in telling faces apart from cars. What you can do is you can take a whole pile of pictures of faces, you can take a whole pile of pictures of cars, and then across multiple scales, meaning different size fragments, and across fragments located anywhere in the image. These are all examples of fragments. You look for which fragments at which scales are best and are most informative across the whole class of faces and cars for telling faces apart from cars and cars apart from faces. So that means that what you want is you want a fragment that is present in as many of the faces as possible and as few of the cars as possible. Okay? And you're doing it at multi-scale, and so you're really finding the best fragments at multiple scales. So it's informative for telling you where the most information is carried in image for doing a particular task. People get that? Question?
So you're talking about fragments being small pieces of the whole.
Right.
But it seems like some of the features that are going to be most important are actually features like, let's say it's distance between eyes, or
right.
I guess that you're saying it occurs in one fragment.
Well, because it's multi-scale. So
yeah.
if it turns out that the best thing is the entire image, this will find that.
Okay.
Right? It's finding the optimal separator given that. And so, in fact, I'll show you in a minute how that plays out actually when you compare different tasks. So,
well, yeah, it struck me that things that may actually encompass the entire image, you know, I mean, what if it's head shape? You know, you're never going to get
right. So there's certain kinds of global descriptors which you might miss. Like, so there's something called shock graphs that you might know about from Ben Kimia's work and Steve Zucker's work. It's basically an axial description. It goes back to Blum's grass fire models. And shock graphs capture this kind of skeletal model of things that you probably wouldn't pick up under this kind of approach. And yet it might be a skeletal model for distinguishing things as the best way because of articulation and other sorts of things. So actually in the lab now, one of the things we are doing is we've got and eventually we'll get it released, a toolkit of about 6 different computer vision models that you can apply to images to measure image similarity across that image set. And then we've been applying that to fMRI data to see which particular models that have different characteristics, capture different characteristics of the object set are best for explaining different responses within the ventral cortex. So you're right, there are some things that won't capture. But I happen to like this for various reasons. So there's some data that Shimon and his students had collected actually suggesting that this has at least some validity when you look at behavioral and electrophysiological data. Does a reasonable job predicting certain kinds of things. So that's the basic model. Just to give you a sense, this is the data set we're using in this particular experiment. For stimuli, you can go download these pictures if you want a bunch of faces. It's actually getting easier to find face sets, but this is still a fairly unique face set in the sense that we have a fair number of non-Caucasians. So it's actually hard to find non-Caucasian faces in most face sets. These are mostly collected in either Europe or the United States. They don't tend to have emotional expressions. And we have, for all of the faces in our set, we have these emotional expressions as well, which can be very useful, particularly if you're looking at generalization. And I will say, our emotional expressions are much better than Ekman emotional expressions. And the reason why is Ekman trains actors to do these expressions. We don't do that because that's not how people make emotional expressions. What you do is you show people movies. And you videotape them while they're watching the movies. And how many of you have seen Zoolander? Do you see that? And do you remember the scene, the gas station scene? So we show people the gas station scene, and they crack up, right? You know, and I forget what we used. There was a horrible TV show called Fear Factor. Get great disgust from Fear Factor, okay, and so on. And and actually, this was Alice O'Toole, University of Texas Dallas's idea. But it really works really well. So we get much better emotions. The one problem with the set is that they're relatively low images because this is pre-HD video cameras. But the resolution isn't that critical. You can tell what they are. You know, they're fine. And so if you're interested in about, I think it's like 300 or 400 faces at this point, you can get lots of races and lots of expressions and use them. And that's what we used for this set, and they're downloadable. So, and if anyone wants the protocol of all the videos we use, I can send you that too. So, and well, even better for emotional expression, these are static. If you were to use like 2-second clips, the affect is much more transparent. Emotional expression is really a dynamic property. People use static. But so for someone who's really interested in affective facial processing, that's what you should probably be using. So anyway, that's a vague advertisement or digression. But OK, so here's what we're doing. And this relates back to your question about the task issue. We can define 2 different tasks. We can talk about the detection tasks. So now we're talking about faces versus a whole class of other objects. Like, not class, a whole group of other objects, various other kinds of objects. And these are the features within faces that were found using the mutual information model that were most diagnostic for telling faces as opposed to all the other objects in the set. Okay? And they're relatively small. Okay? And these are very high diagnostic ones. These are actually fragments that were found to not be that informative, but were matched in terms of area to these up there. So these were informative for telling faces versus objects. These were not so informative for faces versus objects. And it's kind of consistent with what we know to be true, that eye regions are better than chin regions and so on. Okay, but this is for the detection task, just telling faces versus that whole thing. But you can redefine the task. You can make an individuation task. So now we're going to ask a slightly different question. We're going to ask, what regions of the face, what fragments and faces across a whole set of faces are most informative across all the faces for telling one face in the set from any other face in the set? Okay? So now it's how informative is that fragment across the whole set of faces for being able to tell one face from another face. And that's the individuation task. And what you find here, these are the fragments that were most informative, and it is things like eye region and the distance between the eyes. And they're larger, right? Because they would need to be larger to capture, configure information, which we think is critical in face recognition. And then these are, again, matched fragments that were actually low information for that task. Question?
The recognition here is, especially with the individuation, is that comparing it to known data? Like, okay, we already have a photo of this face. Hey, this chunk matches. Or is that comparing it to a different photo of the same?
We actually did, it's a good point, we used generalization because we had all the different emotional expressions.
Okay.
And so we went across all of them. So when we say 98% accuracy, that was across, they were all frontally aligned. They have to be aligned. So we didn't use the profile views and other things like that.
But it's the same person, different picture.
Yeah, different emotion and things like that as well. So there's more variability probably here than there is there. Right? But we did match the accuracy of these fragments in terms of how well it performed for both tasks. So we were at 98%. So anyway, so this is the particular set of stimuli we used. And what we were really going to do is it's a slightly different kind of neuroimaging experiment. We're now going to look not at faces versus objects. We're now going to look at the difference in terms of high versus low informative fragments for a given task. So there's really two separate conditions here that don't get compared in terms of, we don't ever do a direct comparison between these in terms of subtraction. The two conditions are for the detection task, what regions of the brains are being recruited when you look at high diagnostic fragments versus low diagnosticity fragments. And then in a separate condition, we're going to say what regions of the brains are recruited when you're looking at high diagnosticity fragments for individuation versus low diagnosticity fragments for individuation. Okay? So that's the comparison now. So the brain maps you're going to see are really separable for this set of comparisons and for this set of comparisons. And it was a very simple task that we had people do. They're seeing these fragments. They were doing, I think it was a one-back location task where they're just saying whether the fragment appeared in the same quadrant of the screen as the previous fragment. So they're really passively viewing these other than keeping them fixated for the most part. And the question is whether or not these are recruiting different brain regions because of their differences in informativeness with respect to different tasks. Okay, so what I'm going to do is orient you first. So FFA, this is the right hemisphere again. That's this yellow region here. You get it bilaterally. And this is a group map across a bunch of different subjects. I think 11 subjects. And so you can see there's the typical thing that you would expect to find in terms of faces as compared to objects. But for the two comparisons I just talked about, okay, so here's for detection. That's in the red here. So what you're seeing is, first of all, there is recruitment of the region, the mid-fusiform region for detection tasks, for detection for high versus low fragments in basically the same region as you're getting FFA. And there's a lot of overlap here. We didn't color code the overlap, but there's a lot of overlap there. You're also getting recruitment of an earlier region, which I'm going to talk about a little bit more, an occipital region, which is sometimes dubbed the occipital face area. Again, there's that sort of bad labeling that then imbues it with a kind of magical theoretical meaning that isn't there. But the occipital face area is being recruited for high versus low fragments for detection. And you're actually seeing a little bit of another area in here too as well, which I don't have as much of an explanation for. Now, what about individuation? What you're seeing is you're seeing recruitment of a much more anterior region along the ventral pathway. And that interior region is pretty much in line with exactly a region that's been given the wonderful name AIT, anterior inferior temporal. But it's a region that's been often associated with face processing. Roger Chatel's lab and other labs have identified it as being involved in face processing. So you're seeing an interesting dissociation here between what regions are recruited for fragments that are diagnostic for detection versus fragments that are diagnostic for individuation. So it's as if Individuation is happening in more anterior regions, and detection, it's not literally detection, but fragments that are just more generic for the faces are happening within the more posterior regions. Okay, now this is a parametric analysis, a standard t-test, if you will, general linear model type of thing. It's a typical thing you see when you see an fMRI experiment. These days, that's not what people do in fMRI. They do what's called a linear classifier, or sometimes a multivariate pattern analysis, or MVPA. And basically, an MVPA analysis, and that could be a support vector machine, it could be a perceptron. There's a lot of other linear classifiers that can all be applied. The linear classifiers, it's kind of a little weird because you'll see a lot of brain maps that look like this, where they're using MVPA to generate the maps, and people misread them all the time because they see the spots and they think, oh, that's the area where you had higher activation relative to some other area. I have someone shaking their head in pain. But I've seen pictures published in like the New York Times where it was an MVPA analysis, but of course they do this. And it's just, but I think there's toolkits now for doing MVPA, and the problem is you can run the toolkit but not understand the process. What MVPA is telling you, quite simply, is where is the most information carried that's contrasting between those two conditions. And information can take, as anyone who just studies neural coding can know, can take all sorts of forms. It could be very sparse, it could be distributed, it could be, but it's where is the most information carried. It's not where is the highest summed response. Am I right? Okay, good. Making sure you guys are the experts. And so we did an MVPA analysis here. Okay, I'm going to talk first about the graph on the left. The graph on the left is the parametric comparisons I just showed you as a brain map. And I'm not showing you the MVPA analysis as a brain map so that people don't misinterpret it. Okay, but this is exactly what I just showed you. That detection is recruiting the fusiform area and the occipital area. And we actually, for the regions of interest here, we don't have the AIT plotted here for this analysis. For complicated reasons that aren't worth getting into, but mostly because we weren't able to localize it across all of our subjects in a standard face versus objects localizer. So we didn't have a good region of interest. If we take the neuroanatomical region, all the effects are there, but we didn't have a way of defining it using a separate functional localizer. So these are all regions: fusiform areas, occipital areas, superior temporal sulcus, left and right. And again, what I showed you before already was the right fusiform and the right occipital recruited for detection, but not the individuation comparison. But look at the pattern analysis, and if you care about the details, they're down here. It's a linear SVM. Okay, and we're doing some PCA up front, and we can debate the merits of doing PCA up front if you want later on. But Adrian, my student, loves doing PCA on everything. But what you can see is a very different pattern emerges in these same regions of interest. It turns out that even though right FFA, in terms of summed activation, is showing no significant difference between high and low diagnostic fragments for individuation, it's carrying huge amounts of information about it. Okay? And in fact, it's the only region of all these comparisons, these 10 comparisons, that's carrying any information about the tasks. Okay, so you're learning something different.
Sorry, on the left panel, is that, I think the number of samples you have 11, is that enough to say that p-value is significant for a t-test?
This is all corrected for the number of comparisons and all the other things you'd want to do. Yeah, it's highly significant. That's what the star is showing you. The stars are the two significant effects. But there's multiple ways you get power. It's also number of observations within your subjects. So we actually have a lot of observations for our subjects here, right? So you can get power by running a lot of subjects or by running a lot of trials.
Okay.
Yeah.
So it's not just 11.
No, the N11 is N subjects.
Subjects,
Yeah. Right. Yeah. So anyway, so from an information point of view, you're seeing a different picture than what you're seeing from a parametric analysis point of view. So what does that mean? How are we doing on time? Okay, perfect. So and this is where I'm going to get much more speculative. But so first of all, we can say with some confidence that the information diagnostic for face detection and for face individuation is differentially coded with respect to this brain network. We can also say with some confidence that the larger identification relevant features or configural features, if you want to call them that, are built from these smaller detection features. And what I would say is that FFA is really the first step in this process. Again, it's sort of unsupervised learning of these individual features of the exemplars, of all the individual exemplars in the set, but somehow they have to become assembled. And what I really think is happening is that you're getting this kind of distributed response with respect to generic features, which is why you get this good detection response when you look at parametric analysis. Because again, fMRI is doing sorry, not fMRI. When you're doing a parametric analysis, you're looking at the sum response, and all of the voxels in that region have to have some difference in that condition to get a significant response for a given region. So what you're really seeing is with respect to these generic features in terms of detection, you're getting actually some kind of distributed code across all of FFA. On the other hand, there is a lot of information carried in the FFA, but it must be very sparse. Because if it wasn't sparse, it was distributed, and there was a difference in the information code across all the different voxel in FFA, what you would see the effect in parametric analyses, but you don't. You only see it in the MVPA. So it means that some of the voxels are carrying a lot of information about specific features, but not most of them. And so the MVPA picks that up, that there's a sparse code there. So I don't know if I explained that as well as I could have, but it was reasonably clear. So you're actually picking up information you wouldn't pick up from a typical analysis. And then the argument is that these configural representations, which are sort of being sent out of FFA in terms of individual features that have reached threshold and would have to be passed on to say who Mike Tarr is, for instance, get passed over to AIT where it really assembles into the holistic representation, and you say who the individual is. OK, so last thing, let's talk about refining the network. And this is a different technique I'm going to talk about. Remember, I talked about the core face processing network and the idea that there's occipital regions, there's this fusiform region. People talk about STS, and they talk about the more interior regions. Okay, so that's often called the core face network. Well, how can we study that in a different sort of way? Well, recently we've got involved in doing diffusion imaging. And I want to get into all the technical details of diffusion imaging. But basically, diffusion imaging is measuring the diffusion of water molecules within any kind of tissue. You can do it for cardiac imaging, you can do it for brain imaging, and so on. But it so happens that white matter fiber tracts in the brain, water molecules diffuse more rapidly across those white matter fiber tracts than they do across the non-tract regions of the brain. So you actually can use this technique to try and look at connectivity of a sort that you wouldn't see under typical structural imaging. And we're using a particular variation of it called diffusion spectrum imaging. If you care about details, I can talk about them later on. But I think right now, I'm pretty convinced that diffusion spectrum imaging, or DSI, has got a leg up on typical DTI. But that ultimately, that difference will disappear. But we're getting very good tracks. We're getting very good fiber crossings in terms of resolving when two tracks cross each other, where the tracks go. And we're getting very good endpoints, relatively speaking. So that's the basic technique. In Silicon Valley, they love to talk about the secret sauce. OK, everyone has secret sauce. And the secret sauce here is a pipeline that was developed by a group of biomedical engineers in Pittsburgh, which does this high-density fiber tracking. It uses a deterministic fiber tracking approach in something called DSI Studio, which they've actually put publicly on the web. You can go download DSI Studio if you want to collect the data. It's using a really multi-direction, a very complex shell. Like a lot of people for DSI or DTI had used only a 16-direction shell, which meant that the number of orientations you could distinguish was quite small. Here we do 257 different directions. So you can very fine-grained distinguish the different directions that the fibers might be crossing in one another. The scan takes a long time. It's almost an hour to scan one subject, as compared to some DTI scans that are like 15 minutes max. So it's a little more of a pain. And then they have a tracking method that they're using then to then get the actual fiber counts out of there. What we're doing in particular is we're identifying functional regions using fMRI, using those as the seeds to track between every pair of ROIs or regions that were functionally defined. And then we're actually repeating that multiple times to make sure that our data actually is reliable. So in our case, we did 1,000 different seeds per voxel. So you're getting some replicability on that whole thing. But the basic idea is you're taking functional data, you're looking at the connectivity between these functionally defined regions. OK? So one analogy I like to make here is that if we think about cortex, it's fairly uniform across all of cortex, same number of layers. Lots of this is not a perfect analogy because there's some local connectivity on a breadboard, but not perfect local connectivity. But imagine that you've got lots of local connectivity. You can think about white matter fiber tracks as being often long-range connectivity that changes the information flow constraints because of the way it connects up regions that often are not near each other and wouldn't have local connectivity typically. So I think it's quite interesting because for me, it's the first time ever that some kind of structural imaging in humans may inform us about something about function, which would be nice. if we could learn something about the structure of the brain that told us something about function, because I don't think that the typical structural imaging you see really tells us much. So, okay. The particular analyses we're doing here involve the following regions of interest. These are all functionally localized again. We've got the typical mid-fusiform region, which is here. We've got a bunch of more occipital or posterior regions, which I'll talk about a little bit more in a minute, and they've got various labels on them, but the critical thing is that they're all more posterior to the mid-fusiform region. You have the STS region, and then you have the anterior temporal region, which are all meant to be the putative regions of the core face network. And these, again, are functionally defined. Okay, one comment I'll make is I talked about these occipital regions multiple times. There's pretty good evidence if you actually look across not just this one specific study by County Girl Spector, but all of the neuroimaging studies done in face recognition. There's really probably at least 3 distinct functional regions appearing in occipital regions of the brain that are face selective. The interesting thing is you look in most papers and they get a region that's not quite in the region that someone else reported, or they get 2 regions, and they kind of ignore it because they don't know how to explain it. So they'll just sort of say, well, yeah, let's just merge it all to one region and call it the occipital face region. But Callany did this, and other people have begun to do it. There really looks like there's pretty good functional distinctions between at least 3 regions of more occipital areas in terms of face selectivity. You'll see why this is important in a minute. Okay, so here's the kind of data we get back. This panel up here on the left is just showing you the functional regions. So this is more posterior to more anterior. There's STS up there. These were where we seeded everything to look for tracks. The tracks are color-coded reporting what regions of connectivity they are. And the critical thing really in here is you can see the tracks coming into the fusiform region are really coming from all 3 of the occipital seeds and then going from the fusiform region into the anterior temporal region. So you're seeing the kind of connectivity you would expect to see. There's one piece of connectivity that's missing there, which you notice that the STS isn't connected to any of this network. There's as many seeds in and out of STS as there were in anything else. In terms of trying to find tractography there, you never find anything. I'll talk a little bit more about that in a minute. Okay. Okay. So let's look a little bit more specifically at the relation of STS. Here again are, now what we've done just for convenience sake, is we've actually done the thing I just accused everyone else of doing, is I've merged the 3 occipital regions into one functional region, just to look at the tractography in a more simple way. So this is the connectivity from the more occipital regions into the mid-fusiform region. Okay, and then this is the connectivity from either of those regions up into superior temporal sulcus. And this is 5 distinct subjects. And what you can see is STS is not connected by any white matter fiber tracks that we can find into these core face network. And this is just the same data as a bar graph in terms of track counts, in terms of STS connectivity to any of these functional regions. So, and then also to be clear, we've done lots of controls. STS is connected to lots of things, just not these particular regions. So it's not like STS is in some kind of wasteland that we're missing it because of weird imaging artifacts or because of some other kind of structural thing about the brain we didn't understand. It's just not wired up in this way to the core face network. And this is just showing the same data in terms of track counts. So you can see the number of fibers connecting more posterior regions to the mid-fusiform region, and you can see how many track counts they have connected, again, for 5 subjects. So it's pretty striking. All right. All right, good. All right. Now, what about these occipital regions? This is 2 different distinct subjects, but the other subjects show a similar pattern. So remember, we have 3 seeds of functionally defined distinct regions that we think are face selective. And then we're looking to see whether or not there's fiber tracks coming from those distinct regions into the mid-fusiform. And in fact, for this subject, all 3 of those functionally defined regions show significant fiber connectivity directly into the mid-fusiform. This subject actually didn't show one of the 3 functional regions. It didn't show the posterior fusiform selectivity. For the 2 regions that we were able to functionally identify, it showed connectivity. They showed connectivity directly into the mid-fusiform again. And this is again showing you in a bar chart now across all of our subjects what the connectivity is between the 3 regions that we seem to find and how they all connect into particularly the fusiform region, the mid-fusiform. You can see that's always the highest bar in terms of connectivity. And again, we did verification. You look at these 3 regions and you look to see whether there's connectivity to other regions, and there isn't as much. And if you look at other regions, the occipital region that weren't, in the occipital areas that weren't functionally defined as being part of the face network. So for instance, you take a region in here, there isn't connectivity from a region in here into the midfusiform. So it really seems to be isolated to the particular face selective regions that you see in early occipital, having these fiber tracks directly into midfusiform. So now we're beginning to have evidence that there's a kind of structural connectivity to this core face network that people had functionally defined before. I should be finishing up very soon, right? Yeah, okay, good. We're almost done here. This is basically showing the same thing. It's just for more subjects, and it's showing the connectivity in there again and how they're all, the fiber tracks are all connected in, so that's fine. This is really, there's 2 more data slides and we're done. But it's showing you from midfusiform into the anterior temporal regions. And again, beautiful connectivity. So the point of view of the core face network, you're seeing long-range connections that connect midfusiform into these interior regions. And when we do controls, again, you don't see the same connectivity where you wouldn't expect to find it. And then lastly, just to give you a comparison to the congenital prosopagnosic data, if you look at the connections between midfusiform to anterior to AIT, and you look at the 2 fiber tracks that Marlene Berman had talked about with her congenital prosopagnosics, in particular the IFOF, there's a tremendous amount of overlap to the point where you even can't see the green fibers in here because they're completely blocked by this fiber tract. So there's really the pathway that we're talking about that connects midfusiform into anterior temporal regions is probably the same as the IFOF, which is exactly the one that was impaired in our congenital prosopagnosics. Okay, so summing up everything, thanks for your forbearance here. There's strong connectivity between these more inferior occipital regions and midfusiform. Midfusiform and AIT are strongly connected, and it tracks the sort of connectivity we've seen in other studies. There's really striking lack of connectivity between these face selective regions and STS. And again, we can subdivide occipital selectivity into at least 3 distinct regions. And it's consistent with a lot of the other data that we've seen, including apparently some neuroanatomical data on the monkey brain, which I have to dig up, but several people have told me that's the case. So again, this is saying what I've said before. And one critical thing to point out, though, is these 3 regions and occipital regions that seem to be face selective are completely functionally ill-defined. We know we can localize them looking at faces versus objects, but I have no idea computationally why there's 3 distinct regions or what they're doing. So there's a whole thing to study there about what we think is happening. And so this is sort of the summary slide back to talking about some kind of theory. When we want to talk about task-driven domain general network for face recognition, I think we can talk about several things in terms of the different regions moving from more posterior to anterior areas. We can say that occipital regions have at least 3 functional and structurally connected things that are going on. We don't know what they are at this point. It'd be nice to study them in more detail. I think FFA is instantiating features that are very specific to specific exemplars within classes that you learn to tell apart individuals within. I think FFA is necessary but not sufficient for face recognition. And I would argue that AIT is in particular what's really doing the assembly of different local features or the configural information that allows you to actually individuate one member from another member of a class. And that SDS doesn't appear to be connected to this network. And so that's it. I love the quote, so I always put it up there at the end of talks. This is to summarize what we need to figure out how to do someday in our lives. Questions?
So I'll, yeah, let you do the questions.
Yes. That's what, so it kills me that when we did that original birding study, the data wasn't collected in a way that we can go back and do a pattern classifier. And it's such a pain in the neck to get a bunch of birders into the lab to sit still in the magnet for that long. But it's exactly, and we kept trying to figure out some way we could save the old data because in fact, what we should show is just as much information about faces is still carried there for them. It's just because of the summed activation thing. So next time one of the groups that I work with at Vanderbilt or in Victoria or somewhere does a training study, we said, well, you got to do it in a slow event-related way so we can do the pattern classifier and at least show that information is maintained even as summed activation reduces. Right?
Could it be possible that STS is somehow indirectly connected to these 3 regions through another region that may not show up if you're just looking for direct?
Oh, no, it must be. I'm sure it is. through, I'm sure through lots of local connectivity permeating through, and possibly even through a set of tracks that have a mediator.
So maybe there's another region that is
Oh, no.
part of that network.
Well, right. So the interesting thing is that when you do a typical quote-unquote face localization task, you compare faces to objects, and you try your best to localize all the regions that are face selective, meaning that they preferentially respond to faces. STS emerges and all these regions emerge, but no other regions emerge. So you'd have to come up with an explanation, which is completely doable, where a mediator doesn't show that selectivity, even though STS then shows the selectivity again. And so I think there's a lot more to be done on what's going on with STS and why it's recruited in some selective way for faces, but that doesn't have that direct connect. Because the direct connectivity at least makes the story simple about why you see, permeating selectivity through the system, because neurons just talk to the other things they're connected to, right? So it just creates more of a puzzle we need to work on. Any question?
So for my graduate work, I did recordings of face response.
I remember that.
Superior temporal sulcus, so it's kind of way back in my past history. But I was always wondering why it didn't map to where the fusiform, the damage would cause prosopagnosia
Right.
in humans. I even tried to record down there, but nobody's seen any face responsive neurons down in fusiform. Do you have a sense of why?
You mean in the monkey?
In the monkey.
So Logothetis claims he gets it all the time.
Okay.
Doris Chow's work certainly suggests that in the mid-fusiform there's a lot of face selectivity. I mean, she finds face selectivity everywhere, though. The brain has become so promiscuous for faces in her case. I believe it, but it's just certain conditions.
So you and the recording guys have to go back down there and really...
Right, and Parrot did too. Some of Parrot's work was in that region. I think so.
I don't even remember. I'll have to go back and look.
Yeah, I'm pretty sure. But there certainly are some, but there, the homologs between the monkey, no one's even really done the simple neuroimaging study in the monkey where you have the monkey look at a lot of faces and a lot of objects and just try and localize fusiform. There's a sort of mixed version of it, but it's not a definitive result for multiple reasons having to do with the tasks and other things. But you'd like to even just start with that. It seems like that'd be low-hanging fruit. And then wherever you see selectivity, from a functional point of view, go and record in those regions. And Nikos was supposed to do that, but then he got interested in all sorts of other stuff other than doing what he'd been sort of previously doing. And he's the one guy that could clearly do it trivially, given the facilities he has. So yeah, I think it's a really interesting question.
But can amnesics learn Greebles to the same level as non-amnesics?
Amnesics or prosopagnosics?
Amnesics, specifically people who have probably damage to temporal lobe areas like the hippocampus.
I have no idea. I have to Google Greeble amnesic and see if someone's done them. But I would guess they'd be impaired on Greebles just like they are like any other domain that they happen to be trying to learn that they're amnesic for. I don't think there'd be anything particularly unique about Greebles. But I mean, some amnesics are very good at perceptual learning tasks.
Yeah, okay, the reason I ask, I'm
right.
sorry, I don't want
No.
But I'm always wondering to what extent are these, you could call it top-down, but these connections from, for instance, hippocampus and say, perirhinal cortex or whatever, are critical for driving the responsiveness and like fusiform face area.
Oh, I think it's completely critical. In fact, I think that it's one of the things we've admitted when we talk about the acquisition of proficiency in a domain expertise. I think there's a lot of evidence now, Tim Kern in particular at Colorado has been collecting, and other people have begun to see that the simple fact of naming is a critical component of it. So if you have people get just as much exposure, and even to the discrimination level, but they never do learn the names, they're much worse. And it's clear that there's some kind of feedback circuit which has to do with the semantics associated with it, and I think these other regions as well in terms of reinforcing. And the other thing we're seeing is degree of engagement or interest is a critical component, which is what hobbies are, and people become proficient. Like I like to ride bikes. I can probably tell you about a couple hundred different models of bikes. Like I ride bike, I know exactly which model of bike. And it's not like I went out and tried to learn about bikes, but there was a high level of interest for me. So it just sort of emerged over time. So I think there's a lot of top-down signals coming in.
Yeah, I mean, to me it seems relevant because when you construct the models of what's going on in an area like FFA, you can do this sort of bottom-up, how do these features get put together, but it seems some studies would suggest that just as important are these signals coming down from.
Right, and I think that's what, I think to some extent, the intriguing thing is Marlene Berman's congenital prosopagnosic data. Because there's some level of organization happening there, and maybe it's still getting the input from perirhinal and from hippocampus and so on, but certainly some sorts of top-down signals are absent. So, so that's pretty intriguing to me about, and again, we don't know if it's really a normal response. We only know it's normal in the most simple way. But this is really one of the big areas that we've been trying to ask, is if you want to think about the visual system, you have to ask about how everything else that you know about is contributing and when you learn new stuff. So I think that one is that these people are really good at compensating. So I think they're better than you and I at voice and noticing local features. Some of them are just academics, you know. They go through the world kind of confused, you know. But there's one guy I know actually at Vanderbilt, a physicist at Vanderbilt, who's clearly just like that. He's kind of was notorious that when they figured out he was congenitally prosopagnosic, everyone went, aha. But it is striking. But you do hear a lot of stories, and I don't know them as well, but just talking to people who have worked with them more, where it is considered, they really do consider an impairment and a problem because of things like recognizing their family or other kinds of weird social situations. They describe bumping into people. The people are like expecting them to socially respond, and they don't, and then they think they're being rude. So you can survive, but it does, you know, but there's also, look, there's lots of people that we now know are probably somewhat Asperger's. Most of them go to Carnegie Mellon and MIT, you know, but they survive too, but they're very asocial. So I think everything's on a spectrum, including face recognition, and it does affect certain interactions, but.
So with the prosopagnosis, do they have a difficult time learning the Greebles as well?
Well, we only did it with that one fellow SM, and it was taking him a lot longer than a normal subject would have. But he had other brain damage too, because there's no such thing as a pure acquired prosopagnosic. They all have agnosia for other object classes also. So there's all sorts of funky things going on there. So I can't definitively say, but he certainly took longer.
Okay, so if there are no further questions, then I'll remind you that if you'd like some coffee or drinks or snacks and so on, it's in this room right over here. Otherwise, let's thanks Mike. Well,